Bom dia a todos. É um prazer hoje a gente ter o professor Emílio Hirsch. Can I speak in Portuguese or is it okay for you? Professor Emílio Hirsch, né, no nosso seminário das 11 horas, então, da sexta-feira do CRID. O professor Emílio Hirsch é professor de Biologia Aplicada. O professor Emílio Hirsch tem doutorado em Biologia Celular e Molecular. É professor titular, full professor no Instituto de Biotecnologia Molecular. E, por sinal, é full professor e atualmente é o diretor do Instituto. O Emílio tem uma linha de pesquisa vasta dentro de sinalização celular e ele tem uma imensa contribuição, principalmente dentro do contexto de via de sinalização de PI3K, mas também não somente com PI3K, ele estuda muito parte de AMP ciclo signaling, AMPK e outras vias, e coloca, vocês vão ver os trabalhos do Emílio, são trabalhos feitos com biologia celular profunda, aplicada, mas que sempre ele extrapola para um contexto de doença, seja ele diabetes, obesidade, câncer, inflamação, vocês vão ver, né? E eu acho que o de hoje é com câncer, né? Com câncer de pâncreas. Além disso, o Emílio é o nosso consultor do CRID, então ele vem todas as vezes que a gente tem reunião do CRID, e além disso, hoje ele está aqui, então, né, ele tem uma disciplina aqui no programa de pós-graduação da farmacologia, já é a 3ª edição, e ele veio agora aqui para nos dar essa disciplina, tem vários alunos aqui que estão fazendo o curso com ele. Para finalizar, eu estou falando em português porque ele entende português, né, acho que deve ser a 6ª ou 7ª vez que o Emílio vem para Ribeirão, e com o passar do tempo ele foi aprendendo português, hoje ele tem um português ótimo, né? Mas com certeza a palestra dele vai ser em inglês, onde para falar ciência tem que ser em inglês. Agradeço muito, Emílio, por você ter aceito o nosso convite de vir novamente para Ribeirão, para a disciplina, e para ministrar essa palestra. Só lembrando que quarta-feira de tarde a gente vai ter uma outra palestra, terça-feira, 4h30, vamos ter outra palestra que faz parte de uma disciplina da Farmaco também, onde ele vai mostrar um outro tópico super interessante que ele vem trabalhando, que conecta sinalização celular com envelhecimento, morte celular, tem envolvimento de STING e um monte de coisa, né? Obrigado, Emílio.
Zeca, muito obrigado. É sempre um prazer estar aqui no Brasil, eu gosto muito. Estou aprendendo, meu português está tá melhorando. Eu estou vendo que a gente fala sempre em português comigo, estou muito feliz. Tudo bom? Gostaria de falar português, mesmo para minha palestra, mas acho que inglês é melhor, por isso que vou trocar com inglês. Desculpe, mas se você quiser fazer uma pergunta em português, acho que eu posso entender, posso. Vamos ver. Okay, then, sorry, I need to switch to English because sometimes I will be able to speak in Portuguese, but we'll see. So, thank you very much, Zeca. Thank you very much, Thiago. Thank you to all, Ricardo, Professor Fernando, everybody. It's always a great pleasure to be here. And Zeca told you that I spent most of my career, actually the largest part of my career. I started with something different, but then I moved into xenotransduction, and I stayed there for almost 25 years. And I picked one specific xenotransduction pathway, that is the PI3 kinase xenotransduction pathway. So this PI3 kinase pathway is activated by receptors. The students that followed my course yesterday and the days before know better than me now, but we have receptors on the plasma membrane. These receptors sense something in the cell, something outside the cell, and then they need to tell the cell to do something more. And most receptors, like tyrosine kinase receptors, G protein coupled receptors, they all induce the production of a lipid secondary messenger molecule that is called PIP3. PIP3 is never there, but when receptors are activated, PIP3 is produced by these famous enzymes, PI3 kinases. And in the past, I worked a lot with PI3 kinase gamma, that is a GPCR coupled PI3 kinase. And usually when they produce PIP3, they actually trigger a series of xenotransduction events that end up in activation of kinases. And AKT is the first one in the cascade, because AKT can bind this phosphorylated lipid, and it's active, and then AKT can modulate the mTOR pathway. And the mTOR pathway is very important in the control of reactions of cells in regards of changes in nutrients availability. So mTOR is kind of controlling the size of the cell, the production of proteins, and in the end, cell growth, metabolism, and of course they have issues in cancer. The point is that I always love to scare everybody with these molecules, and the point is that we talked now about this lipid here that for friends it's called the PIP3. It has 3 phosphates, but the most important is on position 3. But yeah, the picture is much more complicated. And I have to say that in the last 10 years, more or less, we focused a lot on another 3-phosphorylated lipid that is called PI3P2. It has a position 3 lipid, whatever, and it lacks 1 phosphate compared to this one. So the signaling transduction pathway I was showing before is triggered by this one. But this here, that is so closely related. What is it this doing? And the thing is that the more we studied it, the more we realized that these 2 very similar molecules are actually telling the cells to do very different things. And in the end, there are different enzymes, because PI3 kinase gamma makes this but doesn't make this, and PI3 kinase class 2 actually make this but don't make this. So there must be a reason for having specific enzymes that make different lipids, and this must mean something. And in the end, we decided to focus a little bit more on the enzymes that actually make this other specific lipid called PI3P2. And these enzymes are PI3 kinases because they phosphorylate the position 3 of these lipids, the inositol ring, but they are not necessarily activated by receptors. They're quite different from the class 1 PI3 kinases that you probably heard about that respond to receptors and that were depicted in the picture I showed you before in the very beginning. So they are different. They are usually activated by binding to something, but in the end, we don't know yet. We know that at least these enzymes are pretty old. But not that old. PI3 kinases appear in yeast, but these class 2 PI3 kinases only appear in C. elegans. That has 1, and Drosophila has kind of 2, and we have 3 genes for these class 2 enzymes that make PI3P2 and do not make PIP3. And so some time ago, before the COVID, as we say in Italy, we published this review in Trends of Cell Biology, where we tried to summarize what is known about these 3 different enzymes, and we thought about the book that it's still very famous, The Three Musketeers. I don't know whether it's famous in Brazil as it is famous in Italy and probably in France, as it's a French book. So we put this in the cover and they liked it, and so The Three Musketeers of PI3P2 production. And so we summarized a bit about the three, and I will go quickly on alpha and beta, but I will today focus on gamma. And let me quickly go to alpha, just to tell you that na terça-feira, às 4, vou falar do isso. I am going to talk about class 2 alpha, and because we had a paper in Science very recently, and it turned out to be a very interesting story on mitosis, cell proliferation, but also aging, disease, sting, whatever. We also did some work on beta, and I'd like to just quickly send a quick message on beta, because working with some collaborators, actually helping the group of Volker Auker in Berlin, we realized that really the class 1 is doing something, and the class 2 is doing something opposite. Look at that, for example. So here it's what the class 1 make, and this is what I told you before. PIP3 is important for the activation of mTOR. But it turns out, and Volker Auke had a paper in Science in 2017, that the other one, just one lipid missing, just one phosphate missing, this one is inhibiting mTOR. So very strange. So these two pathways are kind of opposing each other. And we think this is true, and we will see in a minute, because we will stress this with the gamma isoform. But in principle, I tell you this quick story on beta, because I think it's a good example of how, let's say, patients' interpretation of diseases can actually strengthen our understanding at the mechanistic side. And I remember that I was in a commission of PhD that were, defending their thesis, and these people were of different medical, disciplines, because our PhD program is kind of mixed with clinical and basic science, and these people were human geneticists, and they were presenting mutations in mTOR in the brain that actually cause epilepsy. So overactive mTOR means epileptic crisis in patients. So there are patients that are born with some mutations that cause hyperactive mTOR. And they found mutations in many different signal transduction pathways. And this poor student reported, look, everything fits. I have a lot of mutations that tell I'm going to have mTOR active, and that's why the patients have epilepsy. But look, I don't know how to, this is really wrong, because I have a PI3 kinase that is mutated, and we think that this is kind of associated with this epileptic crisis. And the poor student didn't know how to explain this, because everybody thought, as I showed you before, that if you have a PI3 kinase activating mTOR and you have mutations that kill PI3 kinase activity, no more PIP3, no more mTOR. So it should be the opposite. No epileptic crisis, just the opposite. And then I said, look, but this is not a classical PI3 kinase. This is a class 2 PI3 kinase. This is strange. This is different. And I think we can collaborate, and I think we can figure out that this PI3 kinase makes another lipid that is actually controlling mTOR in the opposite way as you think. And this poor student was so happy, he immediately went to his boss. Then we started a very interesting collaboration. And we realized that the mutations found in these patients with epilepsy were all killing the activity of this PI3 kinase class 2 beta. So here is the production of PI3P2, and you see the mutants from the patients do not make this PI3P2. And we use the test to assay the mTOR pathway activation. And in all of these mutants, we always saw hyperactive mTOR and this was clearly showing that this strange lipid was doing the opposite as expected. And then we generated some knockout mice for these specific class 2 PI3 kinase, and we generated also a mouse that carries the same mutation of a patient, a truncation, by the way, so it's easier. And actually, we see that the brain of these mice, there is hyperactive mTOR. This is kind of a marker of mTOR activity, and you see that it's here and there. We see red, meaning that this marker is there to tell that mTOR activity is higher. And then we see even that in heterozygosity we have a higher mTOR activity, as if this production of the lipid is so tinily controlled that even a loss of 50% of the protein is enough to cause issues. And actually, most of these patients were indeed heterozygotes. And then we thought, okay, the mice in the end looked kind of normal, but we thought we probably need to stress that because patients do not have epileptic crisis all the time. They just, boom, by chance, something happens and they get an epileptic crisis. So we stimulated these mice with pilocarpine, that is something that triggers this epileptic crisis. And you see that this is the wild type, and this is the severity score of the seizure. This is the score, and that's the score of the knockout, and even the same with the mouse that carries the mutation that is closer to the patient. So we got the cover of brain a few months ago. And the story clearly says that we have well, it doesn't work anymore, but we have patients with a mutation that is triggering epileptic crisis through a strange, unexpected, counterintuitive negative regulation of the mTOR pathway. But now I want to, sure, please. Yeah, wait. I will try. We have a debate between me and this German collaborator. He thinks there is I'm not sure. He thinks that 1433 is binding, blah, 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 blah. I don't like this model. Okay. No, I know it's YouTube, but we are good friends. We are good friends. So we proposed a different model, and you will see in a minute. So now let's go into the story I want to tell today, that is more cancer and metabolism. And it's a bit more complicated, but in the end, the key issue is this lipid is inhibiting mTOR. And so, you know, we have 3 genes. We now know a bit about alpha. We know a bit about beta. What about gamma? And gamma is, poor guy, nobody cares about gamma because gamma seems to be expressed in very specific locations in the body. It's nicely expressed in the liver, and it's expressed in the pancreas. There is a little bit in the kidney, and so let's say I have another picture saying that maybe there is also a little bit in leukocytes. We'll see. But the point is that it's not everywhere like the other 2 kinases, and we thought, if this is present in the pancreas, can this have a role in pancreatic cancer that is now considered the most aggressive cancer we have, that is quickly becoming the first cause of death, at least in Europe. The first cause of cancer-induced death is unfortunately pancreatic cancer. So everybody is going into pancreatic cancer because there is money. That's simple enough. Okay. And pancreatic cancer, I told you, it's a kind of a rare cancer. Hopefully, okay, it's not one of the most common, but yeah, you see that unfortunately the cancer is the pancreatic cancer is going to be very soon the first cause of cancer in it's not working. The laser stopped working, but no worries, I can use the I can also use this, no worries. And the problem, okay, it's a rare cancer, but unfortunately very lethal. The sad thing is that it's very tough to diagnose. Unfortunately, patients arrive telling him, I have problems, when it's really too late. And it's tough to diagnose. And as you see, the majority of patients that are diagnosed with pancreatic cancer, they're diagnosed when surgery, that at the moment is the only option, is really not an option anymore. And no worries, Eka, thank you very much, but it's perfect. Wonderful. Yes, let's optimize. Okay, great. And then but let's see, no, but you see, you get a sound, so no, it should work. Yeah, great, thank you. Okay, sorry, Zeka, thank you very much. I was just mentioning that unfortunately, pancreatic cancer is a problem because patients are diagnosed very late, and as you see, very few get treatment, and unfortunately the survival rate is a disaster. And at the moment there is not a lot of funding, but it's changing. Okay, so that's why we are so interested in pancreatic cancer. And why is it so tough to address pancreatic cancer? Because it has some peculiarities. First of all, there are kind of metabolic peculiarities. So pancreatic cancer, of course, I'm talking about PDAC, so the cancer that involves the pancreatic epithelium, so not the endocrine side of the pancreas. So the point is that this kind of PDAC is organizing a tremendous reorganization of cancer-associated milieu. So a lot of fibroblasts, a lot of inflammation is occurring. And in principle, these cancer cells start to evolve inside the kind of isolated pockets, isolated by the inflammatory reaction. And this is called dysplasia. So when all of these cells kind of block the cancer from the extracellular environment. And this has two major consequences. The first is that acts as a barrier for drugs. So the drugs don't reach the cancer because they are blocked by these fibroblasts and inflammatory cells. And the second is that the cancer inside has a lot of metabolic stresses, like there are less nutrients, there is less oxygen. So the cancer starts to live in a situation where metabolic weights, nutrients, and oxygen are limiting. And this is probably one of the causes of the fast evolution of the most aggressive phenotypes. And as you see, in the end, they have a lot of hypoxia, autophagy. So these typical things happen in a progressing cancer situation. And one of the ideas that are developed aside of this is that the cells tend to adapt to changes in nutrient availability, and this might actually involve the modulation of mTOR. And another key nutrient that is often driving the development of the cancer is indeed the interaction between mTOR and glutamine. So because glutamine is a kind of a essential amino acid, though it's not, but it's kind of. And the idea is that these two together might be important in cancer progression. And of course, we know quite well that mTOR does not only senses growth factors through the PI3K pathway, but a lot of other things modulate the activity of this kinase, mTORC1 amino acids, so glutamine and lipids, glucose, oxygen, stress, you name it. And then the output of the mTOR pathway is just trying to tell the cell, look, there are a lot of nutrients around. So you can grow, you can make more cell, you can make more cancer. So you can imagine that when these are not highly available, there should be something strange happening. So the cells should actually sense that nutrients are not available, and they might stop protein synthesis, they might stop proliferating, they might stop they might actually enter more autophagy and more lysosome biogenesis. And this, in principle, should act against the cancer. But in the end, this is not happening. So in the absence of all these things, it seems as if the mTOR pathway is kind of active. And the results I'm going to show you in a moment are actually going in that direction, and in going in the direction that probably the pancreatic cancer likes to have hyperactive mTOR pathway, and that probably through this PI3K C2 gamma, we found one of the ways cancer tries to connect the two, and in the absence of these things, keep this up to the level required for cell proliferation. So the point is that let's have a quick recap of how mTOR pathway is modulated by amino acids. There are two ways. Most of amino acids like leucine and other arginine, they are going through transporters and through this huge complex containing RAG A and B, small GTPases. And for the students, you see, there is always RAB, but it's on one different side in the end. Here we're talking about amino acids and not about growth factors. But glutamine has a different way, and glutamine is also controlling mTOR activity, but apparently it doesn't really work through the classical RAG complex, and it seems like it's controlled by another set of small G protein cascades, and the key driver of this is the G protein ARF1. And then, of course, when mTOR is activated, even by amino acids, the key is that these amino acids are metabolized to help the cancer cell growth. And I was same as I was telling you before. They use glutamine to make lipids. They use glutamine to make nucleic acids. They use glutamine to make more amino acids. So something that is needed for cell growth. So let me recap again what I told you. So I told you that PIP3 and the growth factor signaling pathway going to class 1 PI3 kinase, mTOR is hyperactive. And actually, you know, this is the classical insulin effect. Insulin activates PI3 kinase and PI3 kinase activates mTOR because there is a lot of glucose around. And so the cell can use this glucose and can make more amino acids, more proteins, and everything. But on the other side, we have this strange PI3-4P2 that apparently is doing the opposite, and it's inhibiting the activation of mTOR. And so again, we went back to our PI3 kinase C2 gamma and tried to understand if this was somehow related to pancreatic cancer. And what you see here is quite striking. Okay, so first of all, well, yeah, C2 gamma is nicely expressed in pancreatic epithelium. There is a little bit in monocytes and T cells. It's maybe worth checking, but I don't know. There are traces. But if you look at lymphocytes, I think they're almost nothing. But the interesting thing is that we find PI3 kinase C2 gamma actually being downregulated in pancreatic cancer. It fits again the way, because if this is an inhibitor of the mTOR activity, and if the mTOR activity is important for cancer growth, well, cells don't like these inhibitors, and just through cancer evolution tend to switch them off. And the point is that the more aggressive the pancreatic cancer was, the less PI3 kinase C2 gamma was available. And we even found in the genome, the cancer genome atlas, that 10%, and it's not bad. I mean, if you think about 10% of a rare cancer, it's something significant. 10% of the cases reported there showed deletion of the gene. So it's really deletion happening, and selection is clearly keeping it for some reason. So we started to think, okay, this makes sense. We should understand this a little bit better. And then if we crossed mice that spontaneously develop pancreatic cancer because they have hyperactive RAS over there, well, if they don't have PI3 kinase C2 gamma, they die much faster. And this is actually the mouse. That was the patients with no PI3 kinase C2 gamma. The mice, this is dramatic. No PI3 kinase C2 gamma, mice die incredibly faster. We follow the cancer growth by MRI, and well, you immediately see that in a wild type you see some local cancer growth, and in the knockout, it's already everywhere. And a lot of inflammation, but still very, very strong phenotype. And then, of course, this fits the idea that we have an inhibitor and that the cancer likes to get rid of this inhibitor, but we didn't have a proof that this was really going into the mTOR pathway. And so that's why we went back to our cells, the pancreatic cancers, and we put them in culture, and then we analyzed under kind of steady state the activation of the mTOR pathway through the phosphorylation of this marker that is a substrate of the mTOR kinase, A6 kinase. And indeed, as you can see in the knockout where PI3 kinase C2 gamma is missing, we have a lot more of mTOR pathway activation because we can detect more phosphorylated downstream proteins like the A6 kinase. And the potential explanation for the increased aggressiveness of the cancer is really that this mTOR pathway is hyperactive. And we did some more immunohistochemistry in our patients with low PI3K 2 alpha, and bingo again, even in humans, we see that A6 kinase and mTOR pathway is hyperactive. And so that's the key. Gamma inhibits the pathway, but why? That's the question, and that's what I want to address now. Because we were really puzzled by that mechanism proposed by the Volker Auke's lab, and then we thought again. Look, mTOR can be modulated by two big ways: growth factors on one side, amino acids on the other. So the question was, do we get an effect on one of the two specific branches, or is it just controlling everything at the same time? And so Maria Chiara, that did, I think, a very, very inspiring experiment under the supervision of Miriam Martini, that is also the key driver here. So the point is that you can immediately test the growth factor pathway by deciding whether to put or not the serum in the cell culture medium. And if we take, the knockout, well, the mTOR pathway A6 kinase phosphorylation is slightly higher. If we remove the serum, boom, 0. So it seems like the serum branch is not affected because we can still have normal control over this serum. And then we thought, okay, let's think about nutrients and amino acids. And yeah, the easiest is to use glutamine, because you always need to add glutamine to the medium. And we said, okay, let's use a glutamine-free medium, and let's see. And now look at the situation. So this is the normal situation. This is the wild type. The mTOR pathway is activated by the serum. Serum is there. Here we have serum, but we don't have glutamine. Okay, so serum is there, and some mTOR activity is detectable, but this is less because there is no glutamine. The cell senses that serum is there, yeah, but somebody is telling me to grow the serum, but I don't have the material to grow. I don't have glutamine. So the cell senses this situation and tries to break the activation of mTOR. And you see that activation of mTOR in wild type cells in the absence of glutamine is much less. There is something happening. I showed you before ARF. So that's, of course, something we kept in mind. But look at the now what happens in the knockout. There is no downregulation of the mTOR pathway. So it looked as if this PI3K 2 gamma was more involved into amino acid mediated mTOR control than growth factors mediated mTOR control. And then we had a fight with referees because life is always hard. But we ended up with this very, very complicated experiment. And I hope this can convince you as well. In the end, it worked out. The referees were happy. So we divided the cells in kind of three conditions. No glutamine, a little glutamine, but not enough, and which is kind of close to the situation in pancreatic cancer. And the normal amount of glutamine you put, actually more than the amount of glutamine you put in the medium. And then we have wild type, knockout, serum is present. So the wild type tries to reduce the mTOR activity. Now we increase glutamine, and this break is, the cell is not pressing the brake anymore. And then when we have a lot of glutamine available, the cell is not pressing the brake at all, and the mTOR pathway can be fully activated by the growth factor pathway. But the situation in the knockout is completely different. So in the knockout, no glutamine, mTOR pathway is still hyperactive. So it really fits the idea that I showed you before. We have a situation in these cancers that food is not available, but they grow as well. So how they grow? Well, autophagy, we don't know precisely, but there is a lot happening for sure. But the funny thing is that mTOR is inhibiting autophagy, so it's still open. On the other hand, even in the presence of a lot of glutamine, so the activation of the pathway is steady state. And the referee said, but show me that this is just correlated to the activity of the enzyme. And this is quantification of the phosphorylation we see here. So we increase glutamine, and in the wild type phosphorylation goes up, but in the knockout phosphorylation stays always up. And this nicely correlates with the production of 3,4-P2. This was a tough experiment, but you can hope you can see that in the knockout there is no 3,4-P2 that is produced in this context. And even increasing the amount of glutamine, nothing changes. But in the wild type, the black line, you see that with zero glutamine, 3,4-P2 is highly available. And then the less glutamine the more glutamine we put, the less of this break is actioned. Okay, so we have more glutamine, but we have less PI3,4-P2. So PI3,4-P2 is produced when glutamine is missing. That's the situation. So we have glutamine in serum in a wild type, and the mTOR pathway is active and S6 kinase is phosphorylated. In the knockout, it doesn't matter. We have that the pathway is phosphorylated as well. The difference is here, because here no glutamine means there is a lot of PI3,4-P2, and now in the wild type, this PI3,4-P2 is inhibiting mTOR, and activation of the pathway goes down. In the knockout, where this 3,4-P2 cannot be the red line, cannot be produced, mTOR is always happy to activate its downstream effectors. So cancer cells probably tend to evolve so that this inhibitor is gone. And the point is, how is this happening and where? And mTOR is on lysosomes. So we thought it must happen on lysosomes. And so we analyzed the lysosomes with LAMP1. And as you see, in normal condition, we have lysosomes, we have PI3 kinase 2 gamma, and we have PI3,4-P2 red. They are somewhere, but they don't localize together. They're never together. If we put no more glutamine, actually, if we remove glutamine, then they go together. So finally, we have lysosomes that contain PI3 kinase 2 gamma. So it means that it goes to lysosomes, and then here it's producing PI3,4-P2. And so there it goes, no glutamine, PI3 kinase 2 gamma goes there and makes 3,4-P2 on lysosomes. Very complicated regulation of mTOR, but in the end, it's much simpler than that, because we only wanted to look at the glutamine, and glutamine is simple, ARF1. So, ARF1 activity is indeed upregulated in the knockout. So if we remove glutamine, ARF1 should actually be deactivated, because ARF1 is this kind of sensing glutamine and telling mTOR active. But ARF1 is hyperactive in the knockout. So what is actually doing this 3,4-P2 to modulate ARF1? ARF1 is a GTPase. Again, we talk about small GTPase at the course, and you remember that small GTPase cycle from active to inactive, inactive GD, active GT, and then, there must be some of these regulators that sense the lipid P3P2. That's what I told Maria Chiara. And well, she said, it will take a long time to understand because there are a lot of regulators. And then I told her, yeah, but now let's say we have hyperactive ARF, so we must have something that is a gap, so that is deactivating ARF that is not working. And then she said again, no, it's going to be hard. I know there's no, look, I have so many of these. I can't. And I said, please come with a gap for ARF that is modulated by PI3K 2 gamma as soon as possible. And she took that literally, because look at this. This is called ASAP, as soon as possible. And so really, it turns out that ASAP has a pH domain that can bind to PI3P2. The others bind to PIP3, so they're not good. So this was really the good one, ASAP 1. And she really came as soon as possible with the candidate that turns out to actually move from the cytoplasms to lysosomes when glutamine is gone. And so you see that here we have ASAP1 in red, 3,4-P2, and these are lysosomes. And when we remove glutamine, boom, they go together. We see white signaling. So that's, I think, the way PI3K 2 gamma is controlling ARF. Of course, we did some more controls, and then we knocked out ASAP, and of course, we have a hyperactive ARF. And the funny thing is that if we kind of have no PI3 kinase C2 gamma active ARF stays in ASAP knockouts, and if we reintroduce PI3 kinase C2 gamma, nothing changes. That's again who wins at the top or at the bottom. And so ASAP knockout wins. If we reintroduce PI3 kinase C2 gamma, it's still hyperactive ARF in the absence of glutamine. So that's the network. We have something that tells the PI3 kinase 2 gamma to go to lysosomes, make 3,4-P2 to recruit ASAP, which is inhibiting mTOR. And yeah, again, we were asked by the referees to show that ASAP1 really binds 3,4-P2, but we told them, look, it's a literature. No, you should show it again. Okay. And so we showed it again, and these are kind of liposomes with 3,4-P2, and it really binds. When we remove it, we actually mutate the pH domain, then it doesn't bind anymore. So it's really binding there. And clearly, when we modulate PI3 kinase C2 gamma activity, we can modulate ARF1 activity, because here is the PI3 kinase C2 gamma knockout. ARF1 is hyperactive. Then we reintroduce wild type PI3 kinase C2 gamma, and ARF activity goes down. We introduce a PI3 kinase C2 gamma mutant that doesn't make the famous lipid, and the ARF1 activity goes back. So we think no glutamine. The problem is we don't have the slightest idea of how no glutamine is activating PI3 kinase 2 gamma. But at least we know what happens on lysosomes. We know that PI3 kinase 2 gamma is now recruited. It's producing 3,4-P2, and ARF is deactivated, and so mTOR is switched off. Of course, if no PI3 kinase 2 gamma is there, ARF is still able to hyperactivate mTOR, and this hyperactivation of mTOR has a lot of consequences in terms of metabolic control. Usually, hyperactive mTOR tells the cell to become more glycolytic. And indeed, we see that glucose consumption in these cancers is higher, and they also kind of produce more lactate. But I think the most interesting is when we analyze the oxygen consumption using the seahorse. And I think you can immediately see here that the baseline, the oxygen consumption is quite different between wild type here and knockout there. So the baseline the knockout is significantly less oxygen dependent. It's indeed glycolytic. And then when we analyze the spare oxygen response, well, you see that the wild type goes from here to there when glutamine is not available. So glutamine can be used, and I'll show you in a moment, in the Krebs cycle. And so if you remove glutamine in the wild type, the Krebs cycle goes down, then a plerotic effect is reduced. But in the knockout, this is not happening. And so the knockout, again, is not able to modulate oxygen consumption in response to changes in glutamine availability. You see, glutamine can be used in the TCA because glutamine can be converted in alpha ketoglutarate, and this fuels the Krebs cycle and causes oxygen consumption. But apparently, in our situation where mTOR pathway is hyperactive, there is a shift toward the use of the remaining little amount of glutamine towards something different. So you see that we have glutamine can be used in two ways. We can have a carbon footprint, and glutamine is used for ATP production. But we have a nitrogen footprint where glutamine is used to make a lot more of interesting intermediate metabolites. So it's important for the production of amino acids. It's important for the production of nucleotides. So if a cell wants to grow, well, it seems like it likes better to produce more nucleotides and amino acids, and that's why the mTOR pathway is trying to push towards this side rather than that side. And again, because we don't have a break, mTOR pathway is hyperactive, and the little glutamine that is coming from who knows where is immediately driven for something that fuels cancer growth. And indeed, you see that this little glutamine used is immediately thrown in the knockout into something to make the cancer grow. And so you see this glutamine, labeled glutamine, now goes to, the label is missing, but either goes to lipids or proteins. And so clearly is used for macromolecules biosynthesis that can lead to cancer growth. And indeed, I would say that as long as little glutamine can be found, the cancer can grow. But then we thought, let's put the cancer in a super harsh condition that is unfortunately not happening in patients because there are always a little bit of glutamine around. But if we put glutamine to zero, what happens? We thought if the cell doesn't have the break and keeps pushing this kind of anabolic use of glutamine, the cell is probably going to die because it will lack the building blocks to make more cells in principle. And that is more or less the same. So if you see, if we have a little glutamine, the knockout grows faster. But if we actually remove glutamine completely, this effect is reversed. So the cancer with no PI3K to gamma and no ability to inhibit the mTOR pathway in the absence of sufficient amino acids grows less. Of course, it is hard to put a cell in a situation where there is no glutamine at all. So the effect is small, but it's still quite significant. And even in let's say time course, we see the same effect. And so we see that without glutamine addiction to the growth medium, the knockout cells are not very good. The point is that they're not very good because they lack some building blocks. And we thought, let's give them the building blocks. We don't give them glutamine, but let's give them some kind of end products made out of glutamine. And I told you before that glutamine can be used to make lipids. So we gave the cancer cells some lipids, and they started to grow again. You know, lipids are important for membrane production. So when a cell wants to become two cells, they need to make a lot of lipids. And when we give lipids from the outside, they don't care about no glutamine at all, and they start to grow again super fast. So in the end, you know, cancer cells are super plastic, and it seems like these PI3 kinase deficient cancer cells learned how to live in a glutamine low condition, and probably rewired a lot of genes. We are doing now a lot of let's say mRNA-seq, but clearly there is some addiction. We don't know precisely what rewired the whole metabolic response, but the point is that if we kind of block the use of glutamine with this drug GLS, well, we see that the growth is lower. The same is true for the mTOR inhibitor rapamycin. If we give rapamycin to these cancers, they grow lower. But the funny thing is that very often now the idea is to combine drugs together and see if we get a synergistic effect. And it seems like we still don't really understand why, but it seems like if we block mTOR1 on one side and glutamine use on the other, we have a much stronger effect. And this is even true in vivo. We put these cancers in mice, or we also grew them. This is punk human cells put in mouse with no PI3 kinase 2 gamma, or here is endogenous growth. And then we treated the mice with this combination, rapamycin to block mTORC1, and this other drug that I always forget, BPTES, that blocks the use of glutamine. And then you see that if we put them together, we have a quite a strong effect, and such a strong effect in such an aggressive cancer is always good. And that is why the paper got accepted in the end in a good, let's say, gastroenterology journal. And that's the roundup. So we have that PI3 kinase 2 gamma acts as a very peculiar, unexpected break of the mTOR pathway, opposite to the classical PI3 kinases. We think that the mechanism is through the modulation of ARF and ASAP, and we think that this is kind of rewiring metabolic control so that cells can grow faster, can make more amino acids, more nucleotides, and they end up in a kind of an addition, so that when we use inhibitors of the two conditions, the use of or let's say the inability to modulate the sensing of glutamine on one side, and the effect of mTORC1, when we put them together, we found a kind of a stronger problem in this cancer. And so what probably makes this cancer more aggressive in the end exposes some kind of weaknesses that we can exploit. And so the idea is now, perhaps because we have these drugs are approved and we have clinical trials, the idea that we proposed is that probably we can think about indicating the use of these two drugs together for those 10% of patients that come up with a deletion in PI3 kinase 2 gamma. And I'm stopping here, and I want to mention the people that got involved. Miriam Martini's dream team with Maria Chiara, where really the driving force is there, but a lot of work has been done by Federico and many others in the group. And I want to thank you for the kind attention. Português, pode. Luiz. Obrigado. Eu vou responder em inglês. Kind of. Yes. Exactly. Ah, good point. Huh? That's right. Very good point. Absolutely, that's a very good point. We do not ask our pathologists. They have records. We can still do that. Huh? I think it's a very interesting idea. Yes, 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 yes. Of course. Absolutely. Yes, 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 yes. We have not checked diabetes because we thought that here the expression is in the exocrine pancreas, but thank you. Yeah, very good suggestion. Cachexia. That's very interesting. That's right. Exactly. I wish I could answer you because I think that I think that if we find this way of control, it's going to be a very important piece of work. But we have no idea at the moment. There must be a sensor, and you are stimulating me, and I have a last minute slide that Zeka knows because we were discussing this yesterday. These are super fresh data, so they're very preliminary, but we tried to do some proteomics with glutamine and without glutamine in the absence of this. I mean, we see a lot of junk like always. I think that this PI3K doesn't like to be expressed, so we have a lot of chaperones being produced. But the second on the top is this funny thing here. So who knows? It's really, you see, this is a histone, so I think this is. But when we remove insulin, this appears to be a partner. And let me go back to this slide here. This strange partner has something to do with sulfur containing amino acids, but glutamine is important there because glutathione is really made out of glutamine with sulfur containing amino acids like cysteine. And cysteine and glutamine are always kind of exchanged together, and having something that is related to this might also end up in in something related to oxidation, reactive oxygen species. And whether this kinase is sensitive to reactive oxygen species, we don't know yet. But that's maybe one way to see if loss of glutamine increases somehow the oxidative stress because glutathione goes down. Maybe we are on I don't know. Next year, I hope. I can tell you more. As you told, the only things we can say so far is that these things happen on lysosomes where mTOR is, but no idea. We don't know anything about what happens on the plasma membrane. In principle, I would say that these PI3 kinases usually don't go to the plasma membrane. I mean, Tuesday I will tell you the opposite, but in general, they like to stay in specialized areas inside the cell, or kind of endosomal endosomal vesicles. And I think we have ideas that probably C2 gamma goes to the lysosome through RAB7 to the late endosome lysosomal pathway. But whether things happen on the plasma membrane, I don't know. It is possible. I would not exclude it. We had a paper some time ago that was showing again effects on intracellular vesicles. So I'm not sure. But who knows? If it's related to oxidative stress, this will certainly go everywhere. So plasma membrane has to be affected as well. Then you asked about autophagy, and I think this is super interesting because they are clearly controlling autophagy because mTOR is a major controller of autophagy. But mTOR activation is happening when food is abundant. And so mTOR is telling the cell, you don't need to eat yourself. And that is a bit counterintuitive. Okay, in this condition it seems like there is no glutamine, but you also kind of want to block autophagy, so it's very hard to understand. But maybe it's what Louis said, is another tissue that is behaving differently. It's kind of too late, and then yeah. Why not? Very good. Português. Vou ficar aqui. alpha, aging and inflammation, sting. Thank you very much.